In this lesson, we're going to take a look at microbial genetics. Well, as we talk about genetics, it's important to understand that this is the study of inheritance and then also heritable traits. And there's a lot of different aspects that the study of genetics is going to include. So first off, this is going to include a study of the physical structure of the genetic material. So that is basically taking a look at the molecules that are involved. Then we're going to take a look at mutations. We're going to talk about what those are and the effects that they can have on an organism. And then lastly, we want to talk about the transfer of genetic information between organisms. So as we start this off, first thing to talk about is the genome and the genome structure. Well, the term genome is really just a fancy word that refers to all of the genetic information of an organism. So if we're studying the genome structure, we're talking about the molecules that are involved. These molecules are called nucleic acids, and these are very large polymers, really the largest polymers that we have inside cells. And we're really talking about millions and billions of smaller pieces sometimes that are going to be joined together one after another. And these large polymers, they're made up of smaller units, monomers, which we call nucleotides. So if we look at the structure of a nucleotide, the nucleotide has three main components to it. They are always going to have a 5-carbon sugar. We call it a pentose sugar because it's 5 carbons. They are going to have a phosphate group attached to that sugar. And then they're also going to have some type of base attached. Oftentimes we call that a nitrogenous base because it is nitrogen containing. So when we start to take these and we do join them together to make the polymers, we do end up with two different nucleic acids. These are DNA, which is deoxyribonucleic acid, and then we also have RNA, which is ribonucleic acid. Now, if we look at the individual components and we talk about the variations that we can have, we do have the three main components to the nucleotide, the sugar, the phosphate, and the base. And the phosphate doesn't change, but the sugar and the base certainly can. So as we're seeing it on this picture right here, first we've got the pentose sugar, which is being shown here in blue or in purple. And for the pentose sugar, we have two different sugars that we can find in that location. We can have deoxyribose or we can have ribose. And that is really what determines whether we're looking at a nucleotide you would find in DNA or one that you would find in RNA. Because deoxyribose, that is what the D stands for in DNA, and ribose is what the R stands for in RNA. Okay, so all of the nucleotides of DNA will contain the deoxyribose sugar, and all of the nucleotides of RNA will contain the ribose sugar. In that base position, the nitrogenous base position, there are really 5 different bases that we can find there. And those bases fall into 2 categories. They are the purines, which have a double ring structure, and then we have the pyrimidines, which are single ring structures. Well, the purines, if we look at those first, adenine and guanine are our example purines. We refer to them by just the first letter, so we call it A and G. And then the pyrimidines are thymine, cytosine, and uracil. There are a little bit of differences when we talk about DNA versus RNA and which bases we're going to find there. So if you look here, just to sum it up, I'll write it over here. In DNA, we can find A and G. So we have both of the purines are possible in DNA. And then for the pyrimidines, it's going to be T and C. When we're talking about RNA, RNA has both of those purines. So again, A and G, but there's no thymine, there's no T in RNA. So we're going to have a U instead of the RNA, instead of the thymine, and then we're going to have the cytosine still. So there is, between DNA and RNA, there's a sugar difference, and there's also going to be a base difference. And again, the base difference is that you only find thymine in DNA, and you only find uracil in RNA. Okay, so that's the overall structure of the nucleotide. Those nucleotides are ultimately going to get joined together to make those large polymers that we were referring to. So here we see how they're actually going to get connected together. When they do connect together, it is going to be the phosphate group of one that connects to the sugar of the next. So if I just write that here, phosphate connects to the sugar. And when we do this, notice that we end up with this linking of sugar, phosphate, sugar, phosphate, sugar, phosphate, the whole way down. So we call that kind of the backbone of the molecule, and it's alternating sugars and phosphates. Notice that those nitrogenous bases, which are just shown here with their names and in the different colors, those are just kind of sticking out to the side. They are not themselves actually involved in joining together of nucleotides to make these long polymers. Now, they are important though, and they're sticking out to the side here. And what they're going to actually do is they are going to be able to hydrogen bond or interact with bases that are sticking out of the side of another DNA strand or another nucleic acid. So we get specific hydrogen bonding pairs that form when we're looking at the bases. And that's what we're seeing in this picture right here. So if we have adenine and thymine, so A and T can form two hydrogen bonds, and that's shown right here. So I'm just going to draw that like this so that we can remember that we get two hydrogen bonds between A and T. A and U, if they happen to be next to each other, they can hydrogen bond as well, and they are also going to form two hydrogen bonds between them. The last pair that we have over here is G and C. And G and C have a stronger interaction because they can form three hydrogen bonds. So these are what we call the base pairing rules. So I'm just going to write that up here. With the base pairing rules, it's basically telling us which ones of these bases are able to hydrogen bond with other ones. If we look at it as far as how they're actually going to be arranged when we look at our overall DNA molecule, notice that we will have those alternating sugars and phosphate, sugar, phosphate, the whole way down the outside, that backbone, and then sticking out towards the middle of this overall double helix that we're going to have, sticking out is going to be the bases. And a base in this strand is going to be able to match up with the correct base in this strand. And when they do, they will hydrogen bond so that you'll either get two hydrogen bonds or we will get the three hydrogen bonds. Now, we don't have U's when we're talking about DNA. That was an RNA specific thing. So these are the combinations that we can possibly have. We can have G and C, and that's here on the top, or we can have A and T down here on the bottom. If we look at it a little bit more formally or a little bit more put together, here we have our overall DNA molecule. It is a double helix. And so if we point out just some overall characteristics that we do want to understand about the structure of DNA, first of all, DNA, it is a double-stranded molecule. And that is referring to the fact that it's made of two strands that are hydrogen bonded together. So in this picture, the little dotted red lines that you see down the middle, that would be those hydrogen bonds that you would find between the base pairs. Another thing to understand is that the strands face or run in opposite directions from each other. So we haven't really talked about the polarity of the nucleotides and of the individual polymers. But if you look up here, we have a phosphate end, which is going to be the 5' end up here on this one, and the other one, it has a 3' end. And so again, this is the phosphate end. It means that on this strand, you have a phosphate hanging off the end. And then on the other strand that's opposite it, it's a sugar that's hanging off the end. So again, it's referring to the fact that they're kind of running this way. You can think of it as the head of one is matching up with the tail or the feet of the other strand. We refer to that as anti-parallel. This double helix, this double-stranded molecule, it is twisted around into a helical formation, which is what you see right here. And another thing about it, other than viruses, genomes are composed solely of DNA. So that's all the organisms that we know of, whether they are bacteria, fungi, human beings, all the different plants that we can have, they're all composed of double-stranded DNA. When we get into viruses, there's a lot more variety there, and in that case, we will see some that have RNA genomes. But for the most part, we want to think of the genomes being composed of DNA. Lastly, the order of the bases. If you look at the letters that are down the middle of this double-stranded DNA molecule, they're representing the bases. And it's the actual order of these bases that carries our genetic information, which is the instructions that our cells need to make RNA molecules and then also protein molecules. So the order of the bases is extremely important. And the two strands, having the two strands there is also extremely important because these two strands, they are going to allow the cell or enable the cell to make an exact copy of itself. And that's something that's important every single time a cell is going to replicate. They would have to be able to copy all the genetic information so that they have copies for those new cells that they're producing. I do want to point out that the two strands that we do have, they are what we call complementary to each other. And by complementary, that is referring to the fact that they have that opposite base pair in the opposing strand. So if we remember our base pairing rules, basically if we have an A over here, it's always going to be across from a T. If we have a G over here, it's always across from a C, and so on. That would be the whole way down this DNA molecule. So that's the structure of our DNA molecule. The amount of DNA that we actually have in the genome is going to vary very widely. The smallest one that we know of, you can see that right here, this Carcinella. This contains about 160,000 base pairs, so that's what the BP stands for. We call it base pairs because it's always a double-stranded molecule, and it'll always be, one base here across from that opposite base paired participant in the other strand. That's the smallest. If we talk about E. coli, that's a bacteria. That one's got 4.6, that's million base pairs. So with bacteria, thousands and millions are certainly possible. When we get into humans, we can be up into the billions. So again, genome size, the actual amount of DNA that we do have inside of a cell, is very widely variable. If we focus on the prokaryotic genome first and talk about what that arrangement is like inside of a cell, typically prokaryotic chromosomes, there's just one of them, and it's going to be a circular DNA molecule. So when we say that it's a circular DNA molecule, we're referring to the fact that you've got a double-stranded molecule, but it's connected around into a circle, kind of like this. And that then is going to be held inside what we call the nucleoid of this cell. And so you can see that you do have this region inside this cell that's colored just a little bit different from the surroundings. That's the nucleoid. One thing to understand about the nucleoid is that there is no membrane around it. And so since there is no membrane around it, that means that anything that really happens to be in the cytoplasm is going to be very close to the DNA that we have. So it would be able to actually come in contact with the DNA in that nucleoid. Another thing about the DNA that we do have in the prokaryotic cell is that that DNA is folded into large loops, and those large loops are held in place by RNA molecules and then also proteins. So it really is a massive quantity of DNA that we have there, a really large circular molecule. And so if you look here, you can see that you have these long loops that are coming out. This is basically DNA that has been allowed to kind of come out of the cell so that you can get an idea of how big these molecules actually are. So what we're seeing down there is that your DNA forms these really large loops like this, which is going to help to really compact that into that nucleoid region. We have a gyrase enzyme, which is going to help with that. So the gyrase enzyme is going to coil and fold up the chromosome, fold up the DNA so that it is organized and it fits inside of the cell. So one last thing to know here is that while the typical prokaryote has a single large circular chromosome, that is not all of the time the case. There are some exceptions to that rule. So there are some prokaryotes that have many chromosomes, and then there's some prokaryotes that happen to have some additional chromosomes that are linear in their overall structure. So again, there are exceptions to the rule, but for the most part, we want to think of prokaryotes as having a single large circular chromosome. All right, in addition to that single large circular chromosome, there can also be some other smaller DNA molecules present. So these small molecules of DNA, then they tend to replicate independently of the rest of the cell, and we do refer to these smaller ones as plasmids. So in this picture right here, the red molecule that you're seeing, that would be considered the chromosome. That would get wound up, twisted up, looped up into the nucleoid region. And then you've got the little blue circles that are also there in the cytoplasm. Those are the plasmids that we're referring to here. These plasmids, they are also circular, and they're much, much smaller. These are 1 to 5% of the chromosome size in general. Certainly there is some size variation. You can see that in the picture here. But these do replicate independently of the cell. Some bacterial cells will have plasmids, some of them will not have plasmids. Typically, these plasmids contain information to replicate themselves because they are replicating independently, and then they also contain a few other traits, and it's really just a limited number of other traits. Those other traits typically are not essential to their overall metabolic processes, which is why some prokaryotic cells don't even have the plasmids present in them. We have a number of different types of plasmids. So if we just talk about, mention a few of those different types of plasmids, one of them is what we call a fertility plasmid or an F plasmid. These plasmids contain the instructions for conjugation. Conjugation is going to be a process where two separate bacterial cells will actually connect together, and then they are passing plasmids from one to the other. So it is a way to get some genetic information with prokaryotes and with bacterial cells specifically. So if a cell has the fertility plasmid, that means that that cell is able to make that connection, which is for conjugation. It makes a connection, and then it also sends plasmid copies over to this other cell. So that's one type of plasmid. Another one that we can have is called a resistance plasmid or an R plasmid. These tend to contain some genes for resistant to certain drugs and heavy metals. There's also bacteriocin plasmids. These contain instructions to make specific toxins, which we do refer to as bacteriocins. And then the last one we call a virulence plasmid. This one contains the instructions that are going to enable a specific bacteria to become pathogenic. And when we're talking about pathogenic, that means disease causing. So it is going to give a bacteria the capability to produce disease. If we contrast all of this with the eukaryotic genome, eukaryotic genomes are certainly much larger, and they contain two different parts to them. We have what we call nuclear DNA, and then there's also extranuclear DNA. So the nuclear DNA is going to be DNA that's found in the nucleus. So that's really where the term nuclear DNA comes from. And this DNA that's in the nucleus, it is going to be linear molecules of DNA, still a double helix, but they're linear rather than being connected into a circle. And these typically contain many molecules of different sizes. So we have lots of different chromosomes or pieces of DNA. Depending on the specific organism or species that you're looking at, you're going to have a different overall number to these. There's also going to be elaborate DNA packaging, and that means that the coiling, the twisting, the looping that we saw in the prokaryotes, this is going to be taken to a whole nother level. The reason for this is that there's just so much DNA there. It's a much, much greater quantity than what we had with the prokaryotes. If we take a look at some of that elaborate packing, the elaborate packing is going to take place with the assistance of some different proteins. And the proteins are what we call histones. So we can see that right here. So these are special DNA packaging proteins. And these are going to form larger structures, which we call nucleosomes. And nucleosomes are kind of like a spool that we can take, and then we can wrap the DNA around that. And as we wrap the DNA around it, that is similar to the looping and the coiling that we had. So what we're doing is we're basically taking this large molecule, we're wrapping it, which is going to allow us to shrink and compact and really organize that DNA molecule. Once we get it wrapped around the spools, wrapped around the nucleosomes, you can see that then we make an even thicker structure when we do coil it around even more. And what you're seeing in these images is that basically you can see in the first one we have a purple DNA molecule. We've got some green protein there. And we start to do some wrapping, then we wrap it some more, then we coil it even more, and then ultimately on the end we have this super compacted chromosome which contains DNA, but really it contains a whole lot of protein there as well. And this structure is not something that happens all the time inside cells. There'll be a specific period during their life when they do have chromosomes that look like that, but it's highly compacted DNA and protein. If we look at the extranuclear DNA, Extranuclear has to do with the fact that it is not in the nucleus. So this is outside of the nucleus. This is going to be found in the mitochondria and chloroplast. And the DNA that we find in these compartments is very similar to the DNA that we find in prokaryotic cells. The reason for that is that it is believed that the mitochondria and the chloroplast, they did evolve from prokaryotic ancestors. So these were thought to be at one point independent prokaryotic cells that were eaten or swallowed up, engulfed during the endosymbiotic theory, that overall process. And so we're really just seeing the remnants of that, what used to be an independent prokaryotic cell there. So we do find DNA in those compartments. It's similar to what we find in prokaryotes. It's a circular chromosome. We may also, in some cases, have some plasmids included in the mitochondria and the chloroplast. So you can see here that we just have a picture of the mitochondria inside the very internal inside compartment of the mitochondria. You've got circles of DNA there.